Welcome, everybody. Great to see you. This is the second event in a new seminar series. We call it the Innovation Frontier Insider Stories. And the series is part of MIT HEALS, which is the Institute's Health and Life Sciences Collaborative. In these sessions, we are focusing on how lab discoveries in the life sciences translate into world and life-changing products. And I think it's hard to be at MIT and not be aware of just flying off campus into companies. I don't know if it's entirely unique, but it sure is a different kind of ethos from any other place I've been or any place I've visited. So just to introduce who we are in the series. So many of you know I'm Susan Hockfield. I'm the Whitehead Professor of Neuroscience, and I'm a proud member of MIT's Koch Institute for Integrative Cancer Research. I served as MIT's president from 2004 to 2012, and that was a really vibrant area. We had a rapid acceleration of the convergence of biology, engineering, and medicine, and we are full-throated in the midst of that convergence, which is driving many of the technologies that we'll be showcasing in the series. So our speakers in the series are company founders, and they bring their insights to us in the early stages of biotech tech companies. It's a delight to introduce someone today who doesn't really need any introduction. It's our great good fortune to have MIT Institute Professor Robert Langer, otherwise known as Bob. So many of you might not know, but the Institute Professorship is our highest honor for our professors. And there are only 9 of them, and it is really impossible for me to fully describe or even partially describe Bob's accomplishments and accolades. So I'm going to mention only a few that really kind of called out to me as signature components of Bob's history. So he, like all faculty, writes papers with his students and postdocs and colleagues, and those papers have been cited over 475,000 times. Some of us are laggards in that domain. And his age index of 337 is the highest of any engineer in history. But importantly, Bob doesn't just write brilliant papers, he puts his discoveries into practice. His patents have been licensed or sub-licensed to over 400 companies. We're talking impact here. He's co-founded more than 40 ventures, including Moderna, and he holds 45 honorary doctorates and has received 220 awards, including both the United States National Medal of Science and the U.S. National Medal of Technology and Innovation. He's only one of only 3 living individuals to have received both of those honors. And I would say that is just a little sliver from his huge list of spectacular accomplishments. So this is a little different from most seminars. Our goal in the series is to engage you in the conversation. Bob and I are going to kick off today's session with a very brief introduction, simply to get things going and introduce Bob to those of you who don't know him as well as I do. But as most of you know, he's a legendary engineer and entrepreneur. Bob and I are going to start things off with a short conversation, about 15 minutes, and then we're going to devote the rest of our time to your questions. So please, 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 I encourage you, I exhort you to join our conversation with your curiosity. We want your questions to drive the conversation, and it'll also drive the insights we're going to gain from today's conversation. So before we start, let's get an idea of who we have in our audience. So how many of you could raise your hands, are students? Any undergraduates? How about graduate students? And postgrads, postdocs? Great, terrific. And who among you are in a STEM discipline? Almost everyone. The popular world of biology.
Yeah.
Yes. Okay. Anyone here from business? Anyone here from the Sloan School? Great. How about humanities and social sciences? Terrific. Anyway, thank you all for joining us. Okay, we're going to get started, Bob.
Sounds good.
Yeah, excited to do this. So thank you for joining me. It's always fun to talk with you. It's always a privilege. And we're going to begin with your earliest insight that you could bridge engineering and medicine. Now, you know, we kind of take that for granted today, but you were a very early adopter of that thesis, that biology and engineering and medicine could have things to talk to one another about, and that it could really change the face of what medicine had been. And so was there a particular problem or a particular challenge that couldn't be addressed by one or the other alone? I want to know, what motivated you to actually build that bridge, and how did you make the leap from chemical engineering to biotech and biomedicine?
Yeah, well, it's a great question. Let me try to say a few things. I think they're almost different things. How I got involved in medicine, I'd like to tell you that I had it really well thought out, which wouldn't be true. I really, when I got done with my degree, actually from here in 1974, almost every chemical engineer went to work for an oil company. And I really wasn't that excited about it when I went for these interviews. And I kept looking for something that I could do to help people. I thought about teaching, and I got very involved in helping start a local school for low-income kids, and I tried to get a job developing chemistry curriculum. That's what I was doing, and math curriculum, and I couldn't get any place to hire me. And so then I did think about medicine as a way I might, but I didn't necessarily know what I'd be doing. So I went to work at Children's Hospital with a man named Judah Folkman, and I was the only engineer there. But that being said, there are, to the point you're making, I think a great number of ways that I got to see how engineering could make a difference in medicine, and how that thinking might help. And let me maybe just give one example, which I didn't realize right away, but when I was over at the hospital, I was working on materials. So I was curious, how did materials find their way into medicine? And almost everything that was done up until this time was basically a medical doctor going to their house and trying to pick a material in their house that kind of resembled the organ or tissue they wanted to fix. So for example, the artificial heart, which was developed in initial ones in 1967, they picked a lady's girdle because it had a good flex life. And then, like for breast implant, they picked a mattress stuffing. The first one was polyurethane, the other was a foam, and those made progress, but they also caused problems. Right, blood hits the surface of the artificial heart, and it could form a clot, and would go to the patient's brain, and they'd get a stroke and they'd die. So I started thinking, what you learned in chemical engineering was like design. You could ask a question, what do you really want in a biomaterial from an engineering standpoint, chemistry standpoint, biology standpoint, and then try to synthesize it. And so we kept doing a lot of things like that, but there are lots of examples like that.
Yeah. So how did you actually, tell us about how you actually made the match with medicine? So what was your first
well, the big
foray?
So the big project that I worked on, so Judah Folkman had this idea that if you could stop blood vessels from growing, that might be a whole new way to stop cancer. And he had a lot of, I think, pretty, biochemists and other people work on that, but it wasn't really happening. So when he asked me to join, one of the things that we wanted to develop better was what we call a bioassay, a way to study blood vessel growth. Problem with blood vessel studying it is there's always a lot of background blood vessels wherever you go. But there are a couple places where there are none or very few. The chick choriolentoic membrane is one, and the cornea is another. So we had this assay we were trying to do where we'd put a tumor in some of these sites, but then we needed a tiny little nano or microparticle that we could put right next to it to deliver some of the different things we were isolating, all of which were very large molecules. And that hadn't been done before, and people actually didn't think that was possible. He was on the advisory board of ALZA, which was the only company in the country at that time working on advanced drug delivery systems. So that was how I got involved, was trying to develop these little nano and microparticles that could deliver these large molecules, and then using that to see if we could isolate for the first time these blood vessel inhibitors.
Fantastic. So, in biology, fundamental biology, there are, new discoveries every day. Particularly now, I've been reflecting a lot on the accelerated pace of advances in biological sciences. And, and this is true in biotech. And so how do you decide which ones are worth pursuing?
Yeah.
How do you actually decide which is going to be the biology match for some engineering innovation?
Yeah, well, if it's for, well, I think if part of it, if you're looking at academics, that's one thing. I think there, you could have a long timeline, and I think that I'd probably judge it on impact. I think, or perceived impact. I think if you're doing it at a company, you really have to have made some. You're going to get, the road in medicine is long, to try to get a product out. So, and investors are not going to keep putting money in. They get tired, and all the employees and founders get diluted if you have to keep going for a long time. So I think there you have to have it made enough, not only want to have impact, but you've made progress. Like let's say you have some good animal data, and I usually think that if it was something we're going to do, we'd probably have a paper, ideally in a top journal like Science or Nature or Cell, and also you'd want really good patents, that cover what you did. And for me, I haven't always done it for just those reasons. A lot of times, some of the students and postdocs in the lab, they've worked on this for 5-6 years, and they want to see it make a difference in the world. So they want to see that happen. So it would have those elements.
Yeah, that's terrific. So I think for our young colleagues, I have this question for you, which is, I'm sure that through your career there have been many who didn't believe in your thesis or your work. And what did you do you do? Maybe there's never any doubt anymore about what Bob Langer can do, but what did you do to start to kind of keep going and can persevere toward your goals when, there are just a lot of doubters out there?
Yeah, well, a couple of things I think that helped me. I mean, one was when I mentioned Judah Folkman, who was my postdoctoral boss. He had that. A lot of people really criticized him. In fact, the newspapers, the Wall Street Journal, New York Times, other places, they said this. Even Science magazine, they wrote in, I think, 1976 or 7, there was a big article about how blood vessel growth, angiogenesis, was a figment of his imagination. And yet he kept persevering. And I think, and watching him as a role model, that was great, very helpful to me. And I think that was one thing. Second thing, really, is I was very lucky when I was a young professor here. I had two very close friends. I still do, but now they're Alex Klebanoff, who was, and Mike Marletta. And, we'd get together very frequently and sort of trade stories. And I think that they were very helpful in supporting me, and I hope me with them.
Yeah, that's great. Thank you. One last thing, again, I'm thinking about where I was when I was, still a graduate student, a postdoc, and, the things that I didn't know that I kind of wish I had known. But so in retrospect, when you think about your student trainee experiences, what prepared you for company founding? This is something we don't have classes in, right?
Yeah. So let me try to answer that in a couple ways. First, nothing did, but I think a couple of things helped. One, when I helped start this school for low-income kids, I was developing math and science curriculums, but you had to raise money. I mean, they to try to raise money for an NIH grant, that's hard. Trying to raise money for this was super hard because they were, they were poor. Nobody had money, and people weren't going to give that. So I had a, I started working on grants and even trying to help find money anywhere you could. So that was one thing that was helpful. A second thing that was helpful, though, was actually Dr. Folkman's view of believing anything was possible. Now, neither of those were about starting a company, but those are elements that I think are very important, to doing it. Not giving up, keep trying, and believing anything is possible. One thing though that might, if you want to give, if I go to tips about all the mistakes I made, that the very first time we started a company, this was now probably over 40 years ago. Alex Klebanoff, who I mentioned was my friend, he said, "Bob, we should start a company." And part of the reasons we did it is I couldn't get big companies to really develop the things that I was doing, and that meant a lot to me. I really wanted to not just write papers, as Susan was saying. I wanted to see things make a difference, and I thought it could, and yet I got these two large companies actually licensed stuff from us, and started doing it, and after a year or two, they just gave up. So Alex said to me, "Well, we should start our own company." But just to maybe, as not tips so much, but just to illustrate about why I don't think it's anything you can really learn. You have to learn by your failures or by doing. So the first thing is, Alex said to me, "Bob, you have to find a CEO." We were going to stay at MIT. So he says to me one day, he said, "Bob, I was just at this company's multi-billion-dollar company, and there's this guy who's a senior director, and he's telling me," he said, "he's willing to leave his company to be our CEO." And I said, "Gee, that's great, So anyhow, we hired him to be a CEO. Turns out he was getting fired, but but it just shows how, So the other thing, which I also learned, of course, it took me a number of years to learn this, So people say, when you get venture capital, that people, they give quite a bit of money. And everybody says, but you have to be careful because you should have lawyers go over the contracts and everything, and go over. So we did. The lawyer said everything's fine. But then one of the exits that happens for companies, and it could be even a good thing, is the company got bought about 4 or 5 years later, and by another company. And it turns out that when the thing was signed, there's venture capitals put in money, but they have a thing that's called participating preferred shares. It's perfectly legal, but what it means is that if the company got acquired in a certain way, not only would the investors get what they put in, let's say they owned, I'll make this up, 80% of the company, but then they'd get an additional 80% of what's left. So that would mean that there'd be 4% left for everybody else, if in that example. Yeah. And I mean, it's perfectly legal, but I've never agreed to that after that, but you just kind of learn by mistake. Yeah. And by the way, they're perfect. That's perfectly, both of those were perfectly, legal things, but you only learn by, I think, your mistakes.
Yeah. So we're going to now turn to you. I mean, I could talk to Bob. We do talk for hours. But now it's time for your questions, because we really want this to help you figure out, get answers to questions that you have. There are mics on the side, and if you'd use them, that would be really helpful, because the auditorium by itself doesn't have great, acoustics. So please come to the mics, and Bob will do his best to answer your questions. And don't be shy. Don't fall on the stairs. Just tell us who you are, please.
Thank you very much. You adopted this.
Tell us who you are, please.
My name is Luca. I'm a biomedical robotics engineer, currently working in the development of IoT technologies for environmental protection. Not so close to the biotech world anymore. You adopted this paradigm as a chemical engineer of seeing what you could solve, for example, developing materials or working with materials. Did you further in life develop further ways of framing problems, for example, from the business perspective, that you saw recurringly being correct, so that you found principles under which you could break down business problems or technical issues and understand this was pretty much a recurring solution that you found useful in life?
Yeah, well, let me say 2 or 3 different things. I think I understand the question, but I do think that there are certainly recurring things. I mean, and everybody might have a different approach. What I have liked as we've tried to do these things, is like sometimes you develop something, you invent something, and it could be a great product. But what I've really tried to do, both academically and in the companies, is invent, and I don't do it necessarily for a business reason, but I think what you'll see where I'm going is what I'd like to invent is like what I call a platform technology. And a platform technology, as John Maraganore at Alnylam would say, is sort of a plug and play thing. And Moderna is a good example of that, right? You could have mRNA for COVID vaccine, for treating cancer, for treating disease X or Y. And so many things are like that. I mean, gene editing is like that, siRNA is like that, and even genetic engineering is like that. So that I think has been a good thing, in my opinion, to do, because there's a couple things that it enables. One is that, you know, if you just licensed your technology just to Merck, and there's nothing against Merck, they just use it for their drugs. But if you have a company that's developing it, you could use it with Merck for their drug, Pfizer for their drug, AstraZeneca for theirs. So you could have more impact. Secondly, it makes it much easier to finance the company for the same reason. You could, you know, some of the companies we've started, we've had many deals, and those companies give you a lot of money. And that money's non-dilutive on the one hand, and also it helps you. You know, it enables you to maybe build manufacturing things, and you're never going to, in my experience, you're never going to succeed 100% of the time. I remember even the earliest company I started consulting for was Genentech, and that goes back to the 70s. And you know, some of theirs worked, others didn't, and so you have shots on goal. And so if you have a really good technology, you have a chance to succeed. So that's one sort of very high level thing for me. Now, some people don't like that. They'd say, well, you know, you have to have a big product. I agree with that too, but you can, with this kind of technology, you can do both, right? You have a lead product and you have a pipeline.
Great.
Platform. Thank you very much.
Yeah, process versus product. Interesting.
Yeah.
Over here, please. Tell us who you are.
Hi, I'm Thomas. I'm a postdoc here at the Broad. I have a question for both of you. How does the school support you? And on the other hand, how do you feel like the school can support you more? Like, how does that interaction work?
I didn't.
Because obviously you were president.
Yeah.
And you had Mr. Langer there as well.
Oh.
So like, how did that interaction work? And what can the school do to push more people to be this kind of successful, but at the same time, what would you have wished MIT to do more? Or what makes MIT special and allowed you to do what you did?
Do you want me to go first?
Yeah, please, yeah.
So
Yep.
I can even help give an example for you. But so at any rate, for me, I mean, really what's special about MIT, there's several things that are special about MIT, but one of them is, I mean, the biggest one to me has been your students, your postdocs, and your colleagues. So I think that has been, you know, when I look back over the last 50 years for me, having had such amazing students and colleagues to collaborate with, I mentioned a couple, but I could mention more. So I think MIT gives you that, and if they gave you nothing else, that would probably be pretty good. But there are other things. MIT actually, from a company standpoint, since we're talking about, has actually quite a few things that are helpful. I know even when I got started, they had this industrial liaison program that brought companies in. We have a technology licensing office that's very professional. In fact, when I talk with my colleagues and I collaborate with people at you know, the different hospitals, and they always say, well, let's make sure we send the patent through MIT, you know, and so we probably do, usually do. And there's, you know, all kinds of other things now. There's the Deshpande Center, which provides grants, and I could go on and on. I mean, there's a lot of just different features. And I would just add, I still, you know, that if I look at the administration, that one of the things, I'm probably trying to think how to not insult other schools, but
Go ahead.
yeah, well, that's okay. But you know, one of the things, like I remember I was in one meeting, and this place was saying, you know, they put a lot of different schools up on how they were doing on entrepreneurism, and they said that there were 3 schools that were at the top: MIT, Stanford, and this school. And then and I kept thinking, I had so many people I knew at that school complaining about that they weren't doing that good a job. And so I brought that up, but they kept saying, well, we have all these statistics, Anyhow, but at the next meeting, actually the president of that university got up, he said, you know, we have all these statistics, but the difference is MIT celebrates this. They celebrate entrepreneurism, they celebrate you know, innovation, and we don't, you know, and I think you are a great example of that. I mean, in so many ways. So, and others too. So I think that is, you know, a lot of places, I mean, just to go over, because people may not necessarily realize this, even apologize for it. They think there's, and it's not, and it can be apologized for, because sometimes people are so worried about conflict of interest, and newspapers write stories that are, you know, that this is bad and that's bad. But MIT consistently is celebrated in innovation and the people who do it. And, I mean, here's a great example of this, you know, this thing that you know, all brought up for HEALS. So anyhow, so that's just, I guess, part of how I'd answer it.
Yeah. Great, thank you. Over here.
Hello? Sorry, I don't know how to use this. Can you hear me?
I think so, but you have to.
Hi, I'm Saad Mohsen. I'm an undergrad. I do bioengineering, biochemistry. You can tell because like the pain's in my eye. But I'm just curious. I'm a junior scientist, and I've done, you know, a lot of experiments. It's kind of been difficult to kind of find, you know.
I'm having trouble
we have a
hearing.
hard time hearing you. Get closer to the mic.
Is this better?
Perfect. That's
Okay. Right. So I'm a junior scientist. I do a lot of experiments, and one thing I found is that, you know, a lot of times I kind of hit a dead end, or have a lot of ideas and I don't know where to go from there. And I'm curious, like, you know, based on your career, have you found that, you know, there are times where you have to kind of kill your darlings, or know that you've kind of exhausted every option?
can't hear you.
know
I'm
that-
sorry.
We're having difficulty.
can't hear
Maybe
you.
it's the microphone. I don't know if that.
I don't know. I think you need to keep your get really close to the microphone, or come out and shout.
Like this? Okay. I also have a very quiet voice.
You know, another possibility, if you just maybe come close,
Yeah, just-
and then I'll repeat the question
That's a good one.
as I understand it.
Yeah. Go ahead. Yeah.
Not too close.
Yeah.
To stop an idea.
Yeah.
Yeah.
a good
Well,
question.
okay. So his question is,
great
when
question. Thank
when do
you.
you know to stop the idea? You know, I think it's that's a very good question, for which there's not an easy answer to me. You know, and I think it's a little bit different academically than in industry. But you know, I think academically, if you're trying to discover something, you know, you might end up doing it for many years if you feel it's important, and you maybe make a little bit of progress here and there. But if it's really important, I guess I'd keep doing it academically. Industrially, though, that's different. You know, I think you try to think of killer experiments that can tell you, will it work? And that's actually gets into experimental design. Is there an experiment, maybe again in medicine, on an animal? Like we often have thought that doing things on non-human primates, if you can get to that point, you know, and let's say you see really bad toxicity, you know, that might tell you to stop. And again, sometimes things get in humans too, and you get, you know, really bad toxicity or something. So things like that, you have to think if you get that, whether there's some simple way to get around it. You know, so I don't know if I'd give up right away, but it would make me think about it real hard if I started to, you know, see something that wasn't working in an animal model that mimicked humans. By the way, if I saw it not working or causing safety problems in a mouse, I'd be concerned too. But a lot of times something will work in a mouse or be safe in a mouse, and it won't be safe in a non-human primate. That's just one way of thinking about it.
Yeah, great. Over here.
Hello, I'm a postdoc at the Koch Institute.
Again, you have to get close to the mic. Yeah, that would be better. That's easier.
Hi, I'm a postdoc at the Koch Institute. I work with Christopher Nabel. And yeah, the question that I want to ask is, so obviously as a PI, but also as an inventor, and I'm assuming also to some degree as an investor at this point, you come across through many projects, many propositions to study something, even if it can be from your undergrad, from your students, or it can be from someone that has a project. So I'm very curious to know what makes you say yes versus what makes you say no to like go after that specific project. And if that is too generic or too general, maybe what it is something that you couldn't say no to, for instance, because it touches you personally or because it's something that you really feel very strong about.
Yeah, well, so to me, I guess the answer to me is if I think something has a lot of impact, that makes me highly likely to say yes. I mean, an example which we've been very active in, and it has actually led to some companies, even though that wasn't necessarily the intent, was, Bill Gates came to see me about 14 years ago, and with the point like, well, our work certainly affected, millions, maybe billions of people, but could we do things to help the developing world? And, and I, by the way, I wasn't thinking that when I was 77 years old, I'd have this, really big lab with lots of people. But, that to me was very compelling, that we could make a difference in so many lives. And, through that work, we've been in 25 clinical trials, and, and it has led to a number of companies, and that whole foundation's been very, very helpful to a lot of the things that we've done. So that would be an answer. Is all of it's, I suppose, about impact or perceived impact.
Thank you.
Yeah, we've learned how to use the mic. Good.
Yeah, we're going to have
Thank
to
you.
invent a better microphone.
Yeah,
Is it audible?
Yes.
yeah, it's great.
My name is Dia. I'm an undergrad sophomore in bioengineering and political science. And well, it's really, really incredible to be listening to you speak today. I knew of Bob Langer before I knew of MIT. My question for you is sort of disconnected to everything that's been happening so far. I'm just curious, what was Bob Langer like as an undergrad? What were Bob Langer's aspirations? What did you think as a 19, 20-year-old, and how has that shaped who you are today,
Yeah.
if at all?
Yeah, it's a really interesting question, and maybe you'll be surprised by my answer. But I really, at that age, and even after and before, I was such a naive person. So when I was in high school, I was good in math and science, and my father and my guidance counselor said, well, you should be an engineer. And I honestly felt, why would that be true? I thought engineers ran railroad cars, and I couldn't see why that would matter. But anyhow, then I did. I was an undergraduate at Cornell, and I was good in chemistry, and I had a very hard time with everything else. And so you have to pick your major. I picked chemical engineering because I was, So, but then really for the longest time, and by the way, another thing that happened for me is I have this attention deficit disorder. I had tremendous difficulty at being in a class and paying attention. So I would sit there and go into a lecture with like a lot of people in it, and I daydream. And I think so I didn't do very well my first 3 terms. Finally, I got this idea that, going to these big lectures wasn't getting me anywhere. And so I started doing problem sets, which for an engineer is useful. So I ended up the last 5 terms doing great. But at any rate, I didn't really have a very clear idea of what I wanted to do. I just, was sort of struggling and getting through stuff. So now I'm a senior, and I've got a couple job offers, I think running chemical plants in like North Dakota or something like that. And I think, geez, I would feel terrible for the people in North Dakota if I was doing that. I'd probably blow up the plant. I really didn't. I really wasn't. Didn't have it very well thought out. So I figured, well, I should go to graduate school so I can, postpone my decision and learn some more. And so that was kind of how I was when I was an undergraduate. I mean, it was really later on when I was that shaped what I did. Not necessarily even my PhD research, but helping start that school and then doing the postdoctoral work that with Judah Folkman that I mentioned to Susan before. But I really, when I was your age, well, I guess I said it, yeah.
Thank you.
Thank you.
I think this is actually at the right height for me. Hi, my name's Maddie. I'm a PhD candidate in the Global Engineering and Research Center here at MIT. And I'm hoping to spin out my work developing a non-invasive, low-cost diagnostic for diabetes, specifically thinking about the developing world as well. So my question is, when do you think is the right time for a technology to spin out versus like how long should you kind of keep de-risking and incubating something at MIT? And if there's no easy answer, maybe you can comment on the kind of costs and benefits.
Yeah, I'm going to answer that in a second. I was just thinking a little bit more about the last question, and I actually when I look back at it, because a lot of times I've had so many undergraduates come to my office, and they're very, and they say that they have to have it all figured out when they're a freshman or a sophomore. And I always say to them, they don't, in other words. And I think that's important that I'm almost, when I look back, actually, for me, I was probably lucky I didn't have it figured out because I got exposed to things. So, and a lot of times kids, the students feel so much pressure that they have to have their career figured out. They have to have everything figured out by the time they're 18. Mm-hmm. So I just wanted to reflect back on that for those of you who are thinking you have to, I don't think you do. And to your question, when I think about spinning something out, if I were to, I generally have had these kinds of rules, but they're very general, that I like to feel that we've usually have a paper that, at least one paper that we've written in a really good journal, maybe several. Also that we've got a really good patent. Patents, especially in medicine, end up being very important, and investors usually will not fund things most of the time if you don't have a really good patent. So those would, I think if I had those, and finally, and again, it might be a little bit different for what you're doing, but I'd like to have a really credible proof of principle. So a lot of what we've done is therapeutics, and so I'd like to have that proof of principle in an animal model that was credible, or several animal models that were credible. So I might have to think about it differently if it was a diagnostic, but that's kind of how I've generally thought about it, at meeting those criteria.
Thank you.
Sure, and good luck.
Yeah, over here.
Hi, I'm Evan. I'm a postdoc at the Whitehead. Thank you both for having this incredible event.
Yeah, you need to be closer to the
A
mic.
little closer
Take
to
the
the
mic off and hold it. It might be
Yeah,
easier.
it is a little.
It's a little low.
I wonder if that microphone is better. I don't know.
A little bit. I'm going to speak really clearly and loudly.
Perfect, thank you.
That's good.
Yeah.
Thank you both. So you have the distinction of being a very successful academician and a serial entrepreneur, and I wanted to give you the opportunity to elaborate about how you did this. So earlier you mentioned there are certain types of projects that are better for an academic and some that might be better suited to a company. It seems that as the sort of the scale and the influx of capital is sort of really increasing in biology, that a lot of projects seems better suited towards industrial spaces. And so I'm curious how you think about what sort of work is best suited towards an academic environment and how you evaluate when a technology is sufficiently mature. And then I also had a little softball question. Just considering that you've started as many companies as there are at least letters in the alphabet, just from a cursory list, I wonder what the Bob Langer productivity function is. How did you find time to do all this stuff over your career?
Yeah, well, I'll do that question. So just to go back over everything. So the last question was, how do you find time? And I guess, I've done it now for almost 50 years, so it seems like I've done a lot, but really if I've done on the average one a year, it just happens that you know, and I've had, and it's not just me, it's like our students and everybody else. So I think, and it happens naturally, like a lot of times the students have wanted to see the company start because of their own efforts in the lab. And then the first part, just to make sure I understand it right, is, you know, how do you pick what should be in a company and what should be in academia? Well, I'd say a couple of things. Academia, I don't really have a timeline. I mean, you want to make sure that students can get papers and stuff like that. But in terms of really doing something, I mean, you know, it could take many, many years, and that to me is okay. I think if you want to get grants, you probably have to show something. But I don't. I feel it's fine. I'm going to try to make this may be too simple a distinction, but I think of what you do in the lab as research. And I think if you're going to start a company, you need to be ready to do at least some development. In other words, and the distinction is research, you don't know the answer at all. You can't ever, you know, come up with a time frame. At least I can't, you know. I remember one time I got a company giving us a grant. They had some young guy trying to monitor this, and he said, well, he was making this Gantt chart about when you, and this was funding MIT, and he was making this Gantt chart and said, well, will you have this done like on like he said, May 20th, at 11 o'clock, you'll have this finished. I was thinking, like, how could you ever do that? You know, it's like impossible. But so I don't like to have a time limit on things like that. Companies, you probably do want to be able to have a Gantt chart to say, well, I'll have at least make really educated, thoughtful guesses that, or maybe more than guesses, about when you'll be able to establish things, because you're going to have to often create milestones. So I think you have to have those kinds of things. And going back to an earlier question, I think you can do that to an extent if you've published some papers, if you've gotten the proof of principle, say in an animal or something, you know, if there's a diagnostic that might be the equivalent of that you've reduced risk some. So that's how I think about the first part.
Yeah, excellent. Thank you.
Sure.
Hi, I'm Jocelyn Ram. I'm IP counsel at the Broad, and I also have a background in biomedical engineering, also from Cornell. And you kind of already touched on the question that I was going to ask was, what do you see the value of IP in rolling out, you know, a new company? But if you want to expand on that, but also I'll tag on another question of what do you see AI and machine learning, how do you see this changing the research in your lab today and in the future?
Yeah, so, well, first on the IP, yeah, I obviously, I think it's super important. You know, just to tell you a story, you know, even fairly recent, that I remember a group of scientists at Harvard and Stanford, they called me up, they wanted to start a company, and they wanted to get some advice, and they told me what they were doing. They said we published this paper in Nature, and we found all these great things. I said, I would think you, and they were pretty famous scientists too. I said, well, I would think that people would definitely, you shouldn't have any problem. They said, well, we thought so too, but then after we published it in Nature, we wanted to give it to the world and not patent it, and nobody will fund us. You know, I mean, the investors won't do it because they said, look, anybody can do it now. So anyhow, that's on that. So I think. I'm sorry, the first part of the second.
How is AI and machine learning?
How does AI, yeah. So AI, well, I guess the first thing that I always want to point out with AI is it isn't new. You know, AI has been around for 80 years, and a lot actually started, not the very beginnings, but a lot of it was pioneered at MIT, you know, 60, 70 years ago. And so, but for me personally, we've probably used it for about 30 years. And I'll just give an example of a couple companies that we use it then and use it now. But and our lab too. But the earliest company that we started, Nick Gallicatos, Mike Seaman, and I started this company, Transform Pharmaceuticals. And we decided to do a very simple thing. It turns out 50%, and this was in the 1990s, so 50% of low molecular weight drugs have huge solubility problems. And because they have huge solubility problems, they won't work. So what we decided to do, and companies and people would try, they'd add this excipient. Excipients are things that might help it be more soluble, and so forth. They keep adding different. So what we did is we actually set up robotic systems, and so we could do thousands of experiments very quickly. And we would add many different excipients, like not just one, maybe 4 or 5, all kinds of mixtures. And we'd do thousands of experiments in a few days. And then what we'd do is, let's say we had a target solubility, which would be here. Well, maybe 3 out of those 1,000 would be above that line. And you'd start to analyze using machine learning, that's what we would call it, what were the chemical structures that worked and didn't work. And it helps you then design the next 1,000 sets of experiments. And you keep iterating. So we have this graph, and you keep seeing that by the time you get to the 5th or 6th iteration, you get quite a few of them. And that company ended up doing really well. It got a 25x return for investors. Johnson & Johnson ultimately acquired it. More recently, and this is just a related example, Gio Traverso, who was one of our fellows and now a professor at MIT, and we created an intestine on a chip. And the beauty of that is one of the other big problems is if you'd like to have more drugs absorbed orally, so rather than injected. So we were able to almost do the same thing. We do thousands of experiments. We can do them fairly quickly. And then you can keep iterating and finally get ones that work incredibly well. So you can increase the absorption. We found over 100-fold just by going through this procedure. Novo Nordisk just gave the company a $2.1 billion biobucks deal for GLP orally. So the first point I'd make on your question, which is great, is really where AI is tremendous is when you have a lot of good data. And it can just be incredibly helpful. And both of those are examples of that. We're also using it in the lab. Dan Anderson was another one of my postdocs who's now a chair professor at Chemie and at the Koch Institute. And so we're using it in silico to look at literally millions of designs of different lipids for making lipid nanoparticles, which could be useful for delivering mRNA or siRNA or things like that. But there's others too.
Thank you.
Can I just do a follow-up while you're at the mic? Which is the patenting concept versus the sharing concept. And as investigators, as discoverers, as scientists, we're taught to spread it as far and wide as fast as possible.
Right.
For a variety of reasons, and the expectation of having proprietary control through a patent, it would appear that would be those two dimensions would be in conflict in a university laboratory. She's saying no, you're saying yes. So, but I think it's an important question.
I'll give you my.
Yeah, just I think that for this audience where we see both of these realms side by side all the time, what are the conflicts and what are the pathways to resolve them?
Yeah, well, maybe I should let you. But the way I look at it is, so we publish everything everything we do we publish, but we also patent it, and so why is that? Well, so if you publish it, anybody can use it. Also, to me, it's very important for the students because it will help be very good, important for jobs as well. Mm-hmm. But the patent does, and this would just be my own perception of this. Everybody might have a different one. But what I feel when I look back for me, and I don't know if other people feel the same way, I feel like about 99% of the time, if you're not your own champion, nobody else will be. Every so often somebody comes up with a discovery that everybody would want to use. But most of the time that isn't the case. So what I've found is that, we patent everything, and then, it's like the students who worked in the lab or myself who are the champions. And if you didn't have the patent, like I say, you wouldn't get the money, and you would never be able to develop.
Make it into something useful.
Yeah, so you wouldn't have, the Moderna is a good example in that, and of course in the beginning, nobody would fund messenger RNA, including lots of venture capitalists. But then when you do it, and you make an impact, of course, then it seems like, well, now lots of people, of course, academically are working on it, and companies are working on it too in different ways. But I think the concern was when you have that big emergency, what do you do? And that does create, I think, a problem. But I think 99% of the time, if you didn't have the patents, you just wouldn't have those companies. One of the things I gave a class, and I'll tell just one story, and then I'd love to hear your answer, because I think it's a good story. I often start the class that way. I said, there was a 19th century politician that was asked, what were the 3 most important things in history? And his answer was kind of self-serving, but he said, the printing press. This was, like I say, in the 19th century. He said the founding of America, that's maybe the self-serving system. And he said the patent system. I think he said that because, look at all the companies, look at what it's done for America and other places. And that politician was Abraham Lincoln.
Wonderful, wonderful story. Thank you. And one of the things I want to call out is I spent 20 years at Yale, wonderful, wonderful university. But the ethos there is very different from the ethos at MIT. And I think this idea, it's not an idea, it's a belief, a very deeply held belief, that the things we work on can be useful. Yeah. And it's our responsibility, to turn these fabulous discoveries that are often discovered serendipitously because you're curious about something, the things that can help the world should. Yeah. And that's kind of central to MIT. And it's, I would say, quite anomalous in most of the universities. So it's a wonderful place in that way.
Right, and that gets to what I was saying before about MIT, and you celebrated it. Really, I've seen it firsthand. I'm sorry.
Go ahead.
Yeah, and I would just add that patents are meant to stimulate innovation, and it's a limited right for a limited amount of time. And if we didn't have that, like you said, like many of these companies would not be able to have any funding in order to develop all of these life-saving technologies.
That's right. A virtuous circle. Thank you. Please. I'm sorry for jumping in, but I just had to seize the moment.
Yes, of course. Hi, I'm Goya. I'm a graduate student. I'm actually working on the Laboratory of Translational Engineering here at the Broad. First of all, I would like to thank you for being here with us this afternoon. It's a very special talk. Secondly, I would like to ask you, from your point of view, Robert, and of course, Susan, from the point of view of someone who has shaped how drug delivery works, what are your thoughts on what are the key points on drug deliveries that still needs to be addressed in the context of vaccine that targets the mucal immunity versus, in order for them to be as impactful as the one that targets the immunity, the general immunity?
I'm not sure.
I'm not sure I understood the question.
I would like to have your opinion on what are the key challenges that still needs to be addressed in the context of vaccines that targets the mucosal immunity
Oh,
in order for them to not
Mucosal.
be
Mucosal
exactly.
immunity.
yeah. Well, I mean, I don't feel like I'm an expert on vaccines per se, but you know, mucosal immunity. I mean, you really want it to be effective. I think we're actually doing some work. Anna Jakulenik, who heads a group with us in our lab, has been doing quite a lot of work on that with the Gates Foundation. You know, I think that you want to make sure you get high titers. You want to make sure it's safe. You'd like to be able to just do it once for a lot of these things. But those have been, you know, the main things. I mean, we're actually doing some on polio vaccine with the Gates Foundation, and hopefully others. You know, with these she's developing different nanoparticles that you could swallow, and has gotten some pretty good results in different animal studies.
Wow, fantastic. Unfortunately, this hour has sped by. How can it be? We've only been talking for 15 minutes. It's always this way. So, Bob, thank you very much, and thank you to our audience for terrific, insightful discussion. And I just have to thank our colleagues from MIT Heels, Caroline Lowenthal, Liz Kasakov, and Reina Laricchia, who managed all of the logistics for this event. Thank you very much for making this happen, and thank you all for coming. Bob, can't never thank you enough for the inspirational force that you have been for me and for so many people at MIT and around the world. Thank you.
I've been lucky to work with you for many years, so thank you.
Thank you, everyone, for being here.